Welcome back. This is the part 2 of immunology, T and B cell activation. Thank you for watching my other video. And let's get started. So here we have APCs, that is B cell, dendritic cells, Langerhans cells, and macrophages. Two signals are required for T cell activation, B cell activation, and class switching. So for the first thing is T cell activation. So what happens is the APC ingests, there is an antigen that is being presented on an APC. This ingests the antigen and migrates to the draining lymph node. So this part is at the draining lymph node. And then, sorry, T cell activation signal 1. The first signal is here, this is a step 1, step 2, and step 3, and step 4. Okay, step 1 is the antigen getting present on the antigen presenting cell. Next is step 2 is T cell activation. Signal 1. Exogenous antigen is presented on MHC2 and recognized by TCR on TH CD4 positive cell. So this is a CD4 positive cell, and MHC2 for CD4, MHC1 for CD8. It's getting presented on this. And then the third step is proliferation and survival, where you have the co-stimulatory signal via the interaction of B7, that is the CD8086 protein on a dendritic cell, and CD24 on naive T cell. Okay, this is a CD28 on the naive T cell. So CD8086 from the antigen presenting cell gets presented on the dendritic cell, the CD28 on the naive T cell. And now this activated T helper cell produces the cytokines. The DC cell is able to recognize the antigen, and it goes against the virus and kills the virus infected cell. Now coming to the B cell activation and the class switching. The whole T cell activation is the same thing, the MHC, MHC1 two-step thing. First is CD4, CD8 for MHC2 or MHC1. And then you have a CD28 on the naive T cell, and CD80, 86 on the dendritic cell. Now, the B cell receptor mediated endocytosis. This exogenous antigen is presented on the MHC2 and recognized by the TCR on T helper cell. CD40 receptor on B cell binds CD40 ligand, CD40L on TH cell. The TH cells secrete cytokines that determine Ig class switching of B cells. B cells are activated and produce IgM. They undergo class switching and affinity maturation. The TH cell activation as above. B cell receptor is and mediated endocytosis happens, and the exogenous antigen is presented on the MHC2, recognized by the TCR on the TH cell, and the CD40 on the B cell binds to the CD40 ligand on the activated T cell, and the immunoglobulin class switching of the B cells happen. Sorry. B cells are activated, they produce the IgM. This undergoes the class switching and affinity maturation. Okay. Now we have the immunology. Immune response. Now, antibody structure and antibody function. A fragment consisting of light and heavy chains recognizes antigens. Fc region of the IgM and IgG fixes complement. Heavy chain contributes to Fc and Fab regions. Light chain contributes only to Fab region. Fab. That's the fragment antigen binding area. Determines idiotype, unique antigen binding pocket, only one antigen specificity expressed per B cell. Fc, constant carboxy terminal complement binding, carbohydrate side chains confers isotype IgM and IgD. So we have the Fab region and then we have the FC region. So a Fab region is the fragment of the antigen binding, determines the idiotype. So antigen binding pocket, only one antigenic specificity expressed by the B cell. FC is the constant carboxy terminal complement binding carbohydrate side chains, confers isotype IgM, IgD, generation of the antibody diversity, antigen dependent, a random recombination of the VJ or VDJ genes by the RAG1 and RAG2, random addition of the nucleotides to DNA during the recombination by terminal deoxynucleotide transferase, random combination of heavy chains with light chains. So, generation of antibody diversity, antigen independent, and random recombination of the VJ, light chain or VDJ, heavy chain genes by RAG1 and RAG2. Um, Random addition of the nucleotides to DNA during recombination by um, terminal deoxynucleotide transferase, random recombination of the heavy chains with light chain. Okay, generation of the antibody specificity is the antigen dependent, somatic hypermutation and affinity maturation. Variable region, isotype switching is the constant region. So we have the disulfide bonds, neutralization, opsonization, and complement activation. Antibody prevents bacterial adherence. Antibody promotes phagocytosis. Antibody activates complement enhancing uh, opsonization and lysis. MAC. Okay, coming to the isotypes of the immunoglobulin. All of these can exist as monomers. A mature naive B cell prior to the activation express IgM and IgD on their surfaces. They may differentiate in germinal centers of lymph nodes by isotype switching, gene rearrangement induced by cytokines and CD40 ligands into plasma cells that secrete IgA, IgG, or IgE. For B cells, IgE mom and dad mature to plasma cells as they age. Affinity refers to the individual antibody antigen interaction, while avidity describes the cumulative binding strength of all antibody antigen interactions in a multivalent molecule. IgG, the main antibody in the secondary response to an antigen, most common in the serum, fixes the complement, opsonizes bacteria, neutralizes bacterial toxins and viruses. Only isotype that crosses the placenta provides infants with passive immunity that start to wane after the birth. IgG greets the growing fetus associated with warm autoimmune hemolytic anemia. Warm weather is good. IgA prevents attachment of the bacteria and viruses to the mucous membranes, does not fix complement. Monomer in circulation, dimer with the J gene when secreted, crosses epithelial cells by transcytosis, produced in GI tract, Bayer patches, and protects against the gut infections. Giardia, most produced antibody overall, but has lower serum concentrations. Released into secretions and breast milk, picks up secretory component from epithelial cells, which protects the FC portion from luminal proteases, IgM. First antibody to be produced during an immune response, fixes the complement. Antigen receptor on the surface of B cells, monomer on B cell, pentamer with a J gene when secreted. Pentamer enables avid binding to antigen while humoral responses evolve, associated with cold autoimmune hemolytic anemia. There is a J chain here and in IgA as well. Next we have IgD. The mature naive B cells, normally low levels are detectable in serum. IgE binds mast cells, basophils, cross-linked when exposed to allergen, mediating an immediate type 1 hypersensitivity through release of immediate inflammatory mediators such as histamine. contributes to immunity to parasites by activating eosinophils. Antigen type and memory. Thymus independent antigens. Antigens lacking a peptide component, lipopolysaccharides from gram-negative bacteria. They cannot be presented by MHC onto a T cell. Weakly immunogenic. Vaccines require boosters and adjuvants like capsular polysaccharide subunit of Streptococcus pneumoniae BPSV-23 vaccine, thymus-dependent antigens. Antigens containing a protein component, diphtheria, toxoid, class switching, and immunological memory occur as a result of direct contact of the B cells with T cells. Complement system of hepatically synthesized plasma proteins that play a role in innate immunity and inflammation. Membrane attack complex defense against gram-negative bacteria. The CH50 test is used to screen for activation of the classical complement pathway. Activation pathways classic IgG, IgM mediated. General motors are make classic cars. In alternative is microbe surface molecule, lectin manners molecule, or any other sugars on the microbe surface. Functions. Then you have C3b opsonization, C3a, C4a, C5a, anaphylaxis. C5a is neutrophil chemotaxis, C5b to 9 is MAC cytolysis. C3b binds to the lipopolysaccharides on bacteria. MAC complex is important for neutralizing Neisseria species. Deficiency results in recurrent infections. Get nice Big Macs from 5 to 9 p.m. Opsonins C3b and IgG are the two primary opsonins in bacterial defense. Enhance phagocytosis. C3b also helps clear immune complexes. Opsonin to prepare for eating. Inhibitors decay accelerating factor DAF, also called the CD55 and C1 esterase inhibitor, help prevent complement activation on cells. Okay, so now we have the pathway. Here we have the alternative pathway, classic and lectin. So alternative is the C3 on the microbial surface, C3b along with the BB, C4b and C2b. From the classic pathway, you have antigen antibody complex, C1 complex, and then C1 complex or MBL or MASB. All of this acts on the C3 convertase, the C3 lectin pathways, MBL, MASB. Microbial surface, and this is the sugar moiety. Okay, and then C3 convertase. You have C3A, C3B, C5, C5A, C5B, C5 convertase, and then C5B with the help of C6 to C9 in the cytolysis. C3B goes into opsonization. Let's go ahead and see what happens in inflammation. So when you have a C3 which is getting activated into C3A and C3B, the C3A part simulates a mast cell degeneration, and even the C5A also helps in the C5A mediated neutrophil chemotaxis. Histamine increases endothelial permeability. Okay, now coming to the C5B and C6 to C9, they all work on the cytolysis, and then we have MAC membrane attack complex. So this is for the nice area. The lysis. Then we have C3B. It goes into the opsonization. Then you have micro complement receptor and phagocyte. Complement disorders, early complement deficiencies, um, and terminal complement deficiencies. We have the protein deficiencies, regulatory protein deficiencies. So let's see what the protein deficiencies are. That early and terminal. Early is the C1 to C4, terminal is the C5 to C9. C1 to C4, we have increased risk of severe recurrent pyogenic sinus and respiratory tract infection. C3b is used in clearance of antigen antibody complexes, increased risk of SLE. Think SLE early. Terminal complement deficiency C5 to C9, increased susceptibility to recurrent Neisseria bacteremia. Complement regulatory protein deficiencies. C1 esterase inhibitor deficiency causes uh, hereditary angioedema due to unregulated activation of kallikrein. Increased bradykinin characterized by decreased C4 levels. ACE inhibitors are also contradicted as they increase bradykinin. Paroxysmal nocturnal hemoglobinuria defect in the PIGA gene prevents the formation of glycosyl phosphatidyl inositol (GPI) anchors for complement inhibitors such as decay accelerating factor DAF or CD55 and membrane inhibitor of reactive lysis MIRL or CD59 causes complement mediated intravascular hemolysis, decreased haptoglobin globin and dark urine can cause atypical venous thrombosis, Budd-Chiari syndrome, portal vein, cerebral or dermal thrombosis. Important cytokines. Acute, you have IL-1, 6, TNF alpha, then recruit IL-8 and IL-12. Secreted by macrophages. IL-1 causes fever, acute inflammation, activates endothelium to express adhesion molecules, induce chemokine secretion to recruit WBC, also called as osteoclast activating factor. IL-6 causes fever, stimulates production of acute phase proteins. Tumor necrosis factor alpha activates endothelium, causes WBC recruitment and vascular leak. So this is causing cachexia and malignancy, maintains granuloma in tuberculosis. IL-1 to IL-6 TNF alpha can mediate fever and sepsis. IL-8 major chemotactic factor for neutrophils. Cleanup on IL-8. Neutrophils are recruited by IL-8 to clear infections. IL-12 induces differentiation of T cells into Th1 cells, activates NK cells, facilitates granuloma formation in TB. Secreted by T cells. Interleukin 2 stimulates growth of helper cytotoxic and regulatory T cells and NK cells. IL-3 support growth and differentiation of bone marrow stem cells. Functions like GM-CSF stimulates proliferation of eosinophils, basophils, neutrophils, and monocytes. From Th1, interferon gamma secreted by NK cells and T cells in response to antigen or IL-12 from macrophages, stimulates macrophages to kill phagocytosed pathogens, inhibits differentiation of Th2 cells, induces IgG isotype switching in B cells. Increase MHC expression and antigen presentation by all cells. Activation activates macrophages to induce granuloma formation from Th2 cells. Interleukin 4 induces differentiation of T cells into T helper cells, 2 cells. Promotes growth of B cells, enhances class switching to IgE and IgG. Ain't too proud to beg for help. Interleukin 5 promotes growth and differentiation of B cells, enhances class switching to IgA, stimulates growth and differentiation of eosinophils. I have 5 base. Interleukin 10 attenuates inflammatory response, decreases expression of MHC class 2 and Th1 cytokines, inhibits activated macrophages and dendritic cells. Also secreted by regulatory T cells, TGF-beta and IL-12 both attenuate the immune response. Interleukin 13 promotes IgE production by B cells, induces alternative macrophage activation. Interleukin 13 promotes IgE. Next, we have the respiratory outbursts. Actually, let me go back here. We have the hot T-bone steak, IL-1 fever, hot, 2 T stimulation, 3 stimulates B bone marrow, 4 IgE production, 5 A production, and 6 is acute phase protein production. Okay, respiratory burst. It's also called this oxidative burst. Okay, involves the activation of the phagocyte NADPH oxidase complex, that is neutrophils monocytes, which utilizes oxygen as a substrate, plays an important role in the immune response. Rapid release of reactive oxygen species. And then we have NADPH, which plays a role of creation and neutralization of ROS. Myeloperoxidase contains a blue-green heme containing pigment that is sputum. It's color safe microbe. NADPH oxidase superoxide dismutase myeloperoxidase. How do superoxide dismutase superoxide ion is formed? Hydrogen peroxide, myeloperoxidase. No, okay, sorry. So this is in the phagolysosome. What happens is oxygen is taken up by, and then NADPH gets oxidized to NADPH plus, and oxygen is O2 minus. That's the NADPH oxidase. This is deficient in the CGD. So and then we have R2O2 minus, which gets into peroxide. This is a superoxide dismutase. Then the peroxide gets converted to the bleach HOCl, which is the hypochlorite bleach. This is through the myeloperoxidase enzyme. And water and oxygen via bacterial catalase is released, and then the peroxide gets into the neutrophil, and then it gets converted to water through the glutathione reductase. Glutathione is converted to GSSG, which is the oxidized glutathione, and then NADP plus from NADPH from the forms NADP plus. This is from the HMP shunt. Glucose 6 phosphate gets converted to 6 gluco phosphogluconolactone. There is a G6PD enzyme, so this is deficient, then this is affected. When okay, then phagocytes of patients with CGD can utilize peroxide generated by invading organisms and converted to ROS. Patients are at increased risk for infection by catalase positive species, that is, a Staph aureus aspergillus, capable of neutralizing their own hydrogen peroxide, leaving phagocytes without ROS for fighting infections. Biocyanin of B. aeruginosa generates ROS to kill competing pathogens. Oxidative stress leads to release of lysosomal enzymes, interferons, interferon alpha, interferon beta, and interferon gamma. They interfere with both the RNA and DNA viruses. Cells infected with a virus synthesize these glycoproteins, which act on local cells, priming them for viral defense by downregulating the proteins to resist potential viral replication by upregulating MHC expression to facilitate recognition of the infected cells. Also play a major role in activating anti-tumor immunity. Clinical use: chronic HPV, Kaposi sarcoma, hairy cell leukemia, condyloma acuminatum, renal cell carcinoma, malignant melanoma, multiple sclerosis, chronic granulomatous disease. Adverse effects: you have flu-like symptoms, depression, neutropenia, myopathy, and interferon-induced autoimmunity. Now we have immune cell proteins, cell surface proteins, and after this, we'll probably take another break for this video and then finish the last part altogether. Cell surface proteins: T cells, TCR bind antigen MHC complex CD3 associated with TCR for signal transduction. CD28 binds B7 on APC. Helper T cells are CD4, CD40L, and CXCR, CCR5 co-receptors for HIV. Cytotoxic T cells: CD8, regulatory T cells are CD4 and 25. B cells: Ig binds antigens 19, 20, 21. CD 21 for the Epstein-Barr virus, must be 21 to drink it. Bar 40 B, and then MHC2, B7, CD 80 and 60. So NK cells: CD16 binds Fc of IgG, CD56 suggestive marker for NK cells. Macrophages: CD14 receptor for PAMPs, that is the LPS, CD40, CCR5, MHC2, B7, CFC, and C3b receptors enhance phagocytosis. Hematopoietic stem cells: CD34 energy state during which the cell cannot become activated by exposure to its antigen. T and B cells become energetic when they expose to their antigen without co-stimulatory signal. Another mechanism of self-tolerance. Passive versus active immunity. Passive is receiving the preformed antibodies. Active is taking the antigen, exogenous antigen exposure. Onset is rapid and passive, and active and slow and active. Duration: short span of antibodies. Half-life is 3 weeks. Active is long-lasting protection because you have a memory. Examples: IgA in breast milk, IgG maternal crossing in placenta, antitoxin, humanized monoclonal antibody for passive. Then you have natural infection vaccines and toxoid for active. Note: IV, IG, and other immunoglobulin preparations can be administered to provide temporary but specific passive immunity to a target pathogen. In a passive, in combined passive and active can be given for hepatitis B and rabies exposure. Now we have vaccination. This I'll talk about it in the next video. Thank you for watching. Please like, share, and subscribe to the channel. Thank you. Bye bye.